Hello, welcome to today's lecture. In this lecture, we'll be doing primary hemostasis, so let's get started. So, hemostasis is the control of bleeding. So, whenever there's a cut in anywhere around your body, your blood vessels would be open, right? And blood would flow out of the body. Hemostasis is that control of that bleeding. There are two types, there's primary and secondary hemostasis. Primary hemostasis deals with the formation of something called a platelet plug, and secondary hemostasis is something called a coagulation cascade, or our professor, for the people who are my classmates who are watching, our professor mentioned it as clotting cascade, and that is basically the stabilization of that platelet plug. So first, let's start with primary hemostasis. In the next lecture, we'll do secondary hemostasis, which has got to do with intrinsic pathway and extrinsic pathway. So let's get started. So there are 4 steps for primary hemostasis: vasoconstriction, adhesion, activation, and aggregation. First, let's go through vasoconstriction. So as you can see, there is a blood vessel here. You can see the vessel wall, endothelial cells. As we know, blood vessels are lined with endothelial cells. And you can see this is that damaged area where blood will flow out. Now, when there is damage to a blood vessel, there's something called a vasoconstriction. So the diameter of the blood vessel would actually decrease to reduce blood flow, to reduce excess blood going out of the body. So, because of the injury, you could see the endothelial cells also would get damaged. And these endothelial cells would produce something called endothelin and von Willebrand factor, which is VWF. Endothelin is a hormone that is responsible for vasoconstriction because it stimulates the smooth muscle to contract. Okay, so that causes vasoconstriction. Von Willebrand factor is useful for adhesion of platelets, but first let's write down that endothelin is responsible for vasoconstriction. So endothelial cells produce VWF, which is von Willebrand factor, and endothelin, and endothelin stimulates smooth muscle contraction, causing vasoconstriction. Okay? Now let's move on to step 2, which is adhesion. Adhesion is VWF binding to the platelet. So let's go through it. So VWF or von Willebrand factor, which is produced from that damaged endothelial cells. Remember, damaged endothelial cells, they produce endothelin, which is for vasoconstriction, and von Willebrand factor. So that von Willebrand factor, it binds to the exposed collagen, as we saw here. Whenever there's a damage to the blood vessel, there would be an exposed subendothelial collagen. So that produced von Willebrand factor would bind to the exposed collagen. You could see this is the von Willebrand factor. It binds to the exposed collagen, and this mediates adhesion to the platelets. So how does it adhere to the platelets? It adheres to the platelets via a receptor from the platelets called GP1B protein. Okay, this receptor binds to VWF, and that's how platelets adhere around the damaged blood vessel. So this is very simple. Whenever there's a open cut or to our blood vessels, or there's a cut in our arm or our hand or in our legs, endothelial cells would obviously get damaged, right? They produce VWF, which is von Willebrand factor, and that von Willebrand factor binds to the exposed subendothelial collagen. That allows platelets to adhere to the von Willebrand factor using a glycoprotein called GP1B, glycoprotein 1B. And that's how, actually how platelets come around the exposed wound or the cut wound and start the formation of a platelet plug. So, as we discussed, step 1 and 2 was vasoconstriction and adhesion. Step 3 is activation. What is activation? Activation is basically shape change or conformational change of the platelet. Okay, you could see arrow here, shape change. You could see this is the normal platelet, like in a representation of a circle, and you can see there's a shape change, right? these platelets, they do something called degranulate. What is degranulate? Degranulate is release of granules. And these granules, they contain VWF, which is 1 Willebrand factor, which is right here. They contain thromboxane A2, or you could say TXA2, which is right here. And they contain ADP. Remember, ATP is adenosine triphosphate. And when you hydrolyze that, that forms ADP, right? So they produce ADP, okay? So they produce three things: VWF, TXA2 or thromboxane A2, and ADP. Now, VWF, we know that VWF is for binding to the collagen so that platelets can bind to the VWF, right? That's the function of VWF. Thromboxane A2, it helps in aggregation of the platelets, and that's the next step. We'll talk about it. And ADP, the function of ADP is it exposes a receptor on the platelets. You could see exposes a receptor on the platelets called GP2B3A, okay? Glycoprotein 2B3A, okay? GP2B3A, okay? So VWF allows platelets binding around the damaged blood vessel. Thromboxane A2 helps in aggregation of these platelets, and GP2B3A is exposed because of the release of ADP, okay, adenosine diphosphate. And this protein GP2B3A, it allows in aggregation, which is the next step. So before that, let's write down these steps. So 1 Willebrand factor allows more platelets to bind around the wound. Thromboxane A2 helps in aggregation, which is the next step. And ADP allows exposure of a receptor on the platelets known as GP2B3A. Okay? ADP, remember, adenosine diphosphate. And this receptor is very, very helpful for aggregation, which is the next step. So the last step, which is aggregation. If you look at the English definition of aggregation, that is the formation of a cluster, right? A group of something. In this sense, it's a group of platelets, okay? So it forms the platelet plug, the last step of the formation of a platelet plug around the damaged wound, okay? So you could see these are the platelets that are already changed shape and they degranulated. And because of the degranulation, ADP is produced, right? ADP exposes the receptor on the platelets, the GP2B3A. Now, we know that in blood, there are many types of plasma proteins, right? And one of them is fibrinogen, okay? The function of fibrinogen is to bind to the GP2B3A receptor. And this allows, like a bridge, to bind to the other GP2B3A platelet of another platelet. And this allows aggregation, right? It forms a plug or a bridge. And this is called a weak platelet plug, okay? And this is how aggregation actually happens. Fibrinogen in the blood, which is a plasma protein, it binds to GP2B3A receptor and forms a weak platelet plug, okay? And this is basically aggregation and the formation of platelet plugs. So, the next lecture, which is secondary hemostasis, we'll talk about the stabilization of this platelet plug. And that is basically the coagulation cascade or the clotting cascade. So, let's write down before we move on to the next lecture. So, to summarize, primary hemostasis, there are 4 steps, right? Vasoconstriction, adhesion, activation, and aggregation. So we know when there is a wound or when there's an open cut to a blood vessel, hemostasis is the process of controlling the bleeding. There are 2 types, primary and secondary hemostasis. Primary hemostasis is the formation of a platelet plug, which we just discussed, and secondary hemostasis, which is the next lecture, is the coagulation cascade, also known as the clotting cascade, and this is to stabilize the platelet plug, and this is the intrinsic and extrinsic pathways. So the first step, which is vasoconstriction, it damages, whenever there's a cut, it damages the endothelial cells, right? Endothelial cells line the blood vessels. They produce something called an endothelin and VWF, which is 1 Willebrand factor. Endothelin stimulates smooth muscle contraction, which causes vasoconstriction, okay? And for VWF, it binds to the exposed subendothelial collagen. Whenever there's a cut or whenever there's a wound, subendothelial collagen gets exposed, okay? And that VWF, von Willebrand factor, it binds to the subendothelial collagen. And this allows platelets to bind to the VWF by a receptor called GP1B, okay? Glycoprotein 1B, okay? And this allows platelets to adhere around the wound, okay? It starts the formation of a platelet plug. And the third step, which is activation, we said that the platelets undergo something called a conformational change or shape change, and it degranulates, okay? What is degranulates? Degranulates means it releases granules, and these granules contain VWF, more VWF, which is von Willebrand factor, allows more platelets to bind around the wound. It contains thromboxane A2, which helps in aggregation, and it contains ADP, adenosine diphosphate, which allows the exposure of a receptor on the platelets known as GP2B3A, okay? And this is that receptor. This GP2B3A, because of ADP, it gets exposed into the blood, okay? The last step, which is aggregation, GP2B3A, it binds to a plasma protein, which is fibrinogen, and it forms a weak platelet plug, okay? Which is right here. And this weak platelet plug is, it's aggregating. It's bringing all the platelets in the blood around that wound so that it forms that platelet plug, okay? So yeah, that was, I don't know why this got erased. But yeah, that was primary hemostasis. In the next lecture, we'll do secondary hemostasis. So yeah, I hope this lecture helped. And if you have any questions, you could DM me into my Instagram page at dsr.manav at dsr.manav. And yeah, I hope you have a great day. Bye-bye.